This is a TRP operon, and basically this produces enzymes to make TRP. So basically this operon consists of a promoter which has an operator in it, which regulates whether this produces enzymes or not, and then it has a DNA sequence which then produces enzymes. The operator is regulated by a repressor, which is produced by a TRP repressor DNA sequence, which is a bit before the actual operon in the DNA, and that goes through that. An RNA copies the information from that sequence and then produces the repressor. So this operon is initially active, and it becomes inactive when the operator is repressed. So basically, TRP acts as a co-repressor. It binds to the inactive repressor, makes it active, and binds to the operator, and this prevents the RNA polymerase from binding to the promoter and then producing the enzyme later on. This is an LAC operon. It produces enzymes to help break down lactose. Basically, this operon is initially turned off because the repressor is already binding to the operator, which prevents the RNA polymerase from binding properly to the promoter and producing the enzyme, the DNA sequence. Basically, allolactose, which is a component of lactose, binds to the repressor, making it inactive, and then allowing RNA polymerase to go bind to the promoter and produce these enzymes, beta-galactosides, permease, and transactase. This is a CRP operon. Basically, when you have low levels of glucose in the cell, you have high levels of cyclic AMP, and then you have high levels. And so that binds to a regulatory protein for cyclic AMP called cyclic AMP's regulatory protein. This then binds to the RNA polymerase with the LAC operon and helps to produce a lot more lactose enzymes than the LAC operon regularly would. So basically, the LAC operon is regulated by two different types of regulatory processes. One's positive, one's negative. Hi, my name's Anam, and I'm doing RNA splicing model recombinant. So basically, the DNA helicase opens up the DNA strands, and then the RNA polymerase binds to the promoter, which has a TATA box, and then it transcripts the DNA strand to form a free RNA, which has introns and exons, and a TRP operon, which has modified guanine, and it has the dual function of preventing degradation and helping it to bind to ribones. And then it has a poly A tail, which has 50 to 250 adenines, which have the same two functions as the 5' cap, but it also helps lead the chain out of the nucleus. So then you have small pieces of RNA, which we call small nuclear RNAs, which form SNRPs. And this combines other proteins to form a spliceosome. These cut out the introns from the free RNA strand, so that you have mRNA, which just has exons and the 5' cap and the poly A chain. And then you have exon introns right there, and your spliceosomes break back into the SNRP sample and the proteins.